Hello, my name is gunwant purbia and I am from group 316. So here my topic is physics of the nerve impulse, in which the ionic channels. The generation of the impulse in nerve cell is an phenomenological process that does not reveal the molecular mechanism involved. In the absence of the permeant sodium and potassium ions, the high frequency conductance of the membrane equals to zero, suggesting an electrical current. The ionic channels are electric current and ionic channels are estimated to have a several hundreds per 1 mm of membrane surface. The, the conductance of the single sodium channel is 4 into 10 raised to the power minus 12 mho, while the latest single potassium channel is 12 into 10 to the power minus 12 mho. The ion exchange view channels is a source of energy for the propagation of the nerve impulse, with the sodium pump providing the driving force. Ionic channels are highly selective, but it is possible to make sodium channels transport other cations by blocking the potassium channels by tetraethyl ammonium. Table 11.2 presents the relative permeability of the channels to various cations, as here you can see. The ions like lithium, sodium, potassium, and these have different values. Like lithium has 0.1120 and sodium is 0.190, like this. And the cross sectional of sodium channels determined from these data is about 0.3 into 0.5 nanometer per square. There are observed large difference in permeabilities to ions of similar sizes. The sodium channels is tightly sealed by one molecule of tetrodotoxin and cytotoxin, which paralyze nerve conduction. The use of these substances is allowed for determination of the number of channels per unit surface area of the membrane and steady of the gating current. So here's the table, and the Hodgkins and Luxi estimated an experimental relationship between the initial change of membrane potential and the peak value of GNA, and they hypothesized that his change was due to the action of an electric field on the disposition of some ions in the channels. The conductance of GNA is proportional to the fraction of these ions displaced by the field. An average experimental value of Z is equal to 3.5. Theoretical models have been developed to account for the shape of curve of GNA versus lambda and the shift caused by the change of the concentration of calcium ions, which block the channels. The membrane contains active particles capable of being in uh, as, two states A and B, with transition associated with the performance of work in an electric field. In state A, the particle form in a calcium complex with the calcium plus 2, while the state B, it contribute to the passage of sodium ions through the channels. Experiment shows that when the conductance of the axon membrane undergoes change, such as upon the excitation, these are structural changes. These changes include change in light scattering, fluorescence, and difference of the membrane. The properties of individual channels have been studied by Ionic, and the direct proofs for this displacement of the charged particle entering the membrane structure under the action of the field have been obtained by Armstrong and Breeze and Lira. The structural rearrangement of the channel caused by change in external electric field manifests itself in the presence of an additional component of the displacement current called the gating current. Measurements were carried out on an axon in which the internal medium was replaced by a potassium free solution and the sodium ions were expelled from the external solution. The conductance of the channels were suppressed by tetrodotoxin and experiment was as the uh, conductance of channels in the fibers that were hyperpolarized to -90 or -100 MV. A positive rectangular pulse was applied to the fiber and transient current were absorbed at the state of the start and finish of the pulse. Analogous measurements were made for negative voltage pulse to the account for the capacity charging current. The result were summed up algebraically, revealing a small asymmetric component of the gating current that depends exponentially on time and is associated with the particles that open and close the sodium channels. Other facts also confirm this su- supposition. Now, the second topic is the mechanical processes in which the mechanochemical system. The muscular contraction is a well studied mechanochemical phenomena that encompasses various biological processes, including the movement of plants cells with flagella and cilia and mitosis and the meiosis, protoplasm movement, phase particles and tails membrane ribosomes relative to the mRNA in polysome and the mechanoreception. Flagella and cilia are special contractile system of bacteria and the protozoa that provide their motion in a liquid medium. They have a similar structure consisting with a diameter ranging from zero point 0.1 to zero point 0.5 nanometer. The proteins of flagella and cilia have been studied little, but their ATS activity has been established. Uh, this form and frequency of the sine wave beat propagating along the flagellum depends on the ATP concentration. The protein are contractile but the differ from myosin and actin. Model theories of this beating of flagella and cilia involved changes in the conformational state of protein uh, contractile units when the contractile wave reaches them. In flagella and cilia, there is a sliding between subfibril and the arm function, like bridges in the actinomyosin of the muscle, and no details molecule theory accounting for the wave-like beating of flagella and the cilia have yet been worked out. Contractile proteins have also been identified in other systems such as spermatozoa, ATPase protein, spermacin, and bacteriophages tail structure. In the civet Natalia cells, the driving force is generated at the gel so at the gel solution interface and fibrous structure slide relative to the surrounding. Here is the schematic of the structure of cross sectional of the flagellum. Here you can see that this is the central fibril, and this the first one is the subfibril B and the second one is the subfibril A. Here the dots are the secondary fibrils, and the connection between these the peripheral, and these are the arms. E. coli cells have four long flagella that rotate due to the energy released during proton transfer. The basal body of flagella has a wheel made up of 16 protein molecule which rotates due to energy released during proton transfer. The the plasmodium performs active oscillatory motion of the protoplasm containing actin and the myosin, similar form to those of muscles. However, myosin form small oligomers and interact with actin. These thin bundles of filament in the plasmodium appears and disappears, and the protoplasm current is regulated by calcium ions, and the motility of the protoplasm is expected to be similar to muscles movement, but the absence of thick filament contradicts this. Mitosis and meiosis involved movement of protoplasm and the chromosome, with the meiotic apparatus consisting of strands connecting centrioles and the chromosome. Spermatozoa flagella grown out of a chromosome center, and the proteins of the mitotic apparatus is similar to actin. The mechanism of the meiotic apparatus is still unknown. The mechanoreception and sound reception are associated with mechanochemical processes, possibly tied to the mechanical effect on ionic channels in cell membranes. The Penician body is a mechanoreceptor that transform mechanical effects into the nerve impulse, reversing the function of muscle. It consists of a nerve ending surrounded by a capsule with the peripheral zone has 30 close elastic sheath, while the center zone has a 60 tightly packed cytoplasmic layers separated by a cleft, and the modulus of elasticity of the peripheral zone is about 106 gram per centimeter square, and that can discern a mechanical force with a range of 150 to 200 dynes. The maximum sensitivity of the Venetian body is recorded at the frequency of 100 to 220 Hz, and the optimal frequency of the flow of action potential coincides with the value. The intrinsic excitation period of the receptor is 9.6 meter and which corresponds to the same periodicity of the excitation of the order of 100 Hz. The biological mechanochemistry is primarily related to the enzymic activity of contractile and regulatory proteins. Enzymic activity is determined by the conformational properties of their proteins, and the forced conformational changes caused by a mechanical effect must alter its enzymic activity. This is supported by experiments such as myosin ATA's activity is being altered when the myosin undergoes deformation in an hydrodynamic field of a dyno dynamitometer. This is the Panicium body. Here's my topic is complete. So thank you.